Hi. Yeah, thanks for being here. Yeah, I'm supposed to have something written by now, and I'm still missing a page.
Uh-oh.
Well, I'm dealing with tensegrities, and it's not that simple. Like they behave like they behave.
Yeah, I know.
You know, that is sometimes. Yeah, so.
Still need to finish
So
that.
I thought maybe I would listen in today and do some graphing.
Okay, yeah.
I just fiddle with the graphs, get them. Well, I need to put 3 of them on like one graph. Yeah.
Yeah.
See if I can do that because then I can show the differences in slope. Right now I'm dealing with a slope problem. And a couple other things. It's strange. You put a stronger attachment holding the model down, and sometimes the slopes increase as you increase the pre-strain, and sometimes they get smaller. They can do both.
Yeah. Wow, that's interesting. Does it depend on where you put it in the network, or?
Well, that too. I tried taking the attachments off the middle, and the thing becomes basically a straight line.
Okay. Yeah.
So you have to have some sort of gradation of attachments. But I tried to make a model that would do that, and it won't attach to the attachments I made in between. Like you make your grid work, and then you try to attach in between, and no, won't do that. It does not compute. This is COMSOL for you. You think you can do something and you can't.
I don't know.
So I couldn't really investigate that much. Like I can mess around with the middle attachments, but that's all. So maybe that's what I should explain today. I need to explain things to a pair of professors who want me to be done yesterday.
Oh.
So what would you do in this case?
Yeah, I don't know the answer, I guess. I mean, you could play around and see, like, if you make stronger attachments in certain locations, if that has an effect on it. Because, dealing with a network, you're dealing with a lot of non-linearities. Sometimes you're dealing with, like, dependencies. So it's kind of hard to, like, isolate it.
It's all intentions. So if you change the attachments in one part, it's going to change the whole system.
Yeah.
So I just there's so many things to investigate with the model, and I've only made it partway through, and then I'm supposed to present it.
Yeah. Yeah, I don't know. It's an interesting problem. I mean, because you could probably you could write an entire thesis on just like evaluating changes.
I modified this little part, and it did all of these things, and then I modified this little part over here, and it did all these different things. Yeah, like you could spend a couple of years just the one model modifying things because it doesn't. I can't automate it, like I said.
Yeah.
So it just takes forever.
That's science, I guess. Thesis land or whatever it is.
Thesis land, yeah. Okay, so I've got Canadian news on in the background. Can you hear that or?
No, it's fine.
No, okay. Right. Anyway. Hi, Dick.
Hi, hello.
Hi,
yes, I can hear it, but I can't understand it.
Okay.
Okay.
All right.
It had something to do with, was it R.F. Kennedy being the head of health and not believing in vaccines and stuff? That's what's on the news. Anyway,
So yeah, good luck with that project.
yeah. I don't even know how to explain it to them. Like, I can only investigate part of this model, and this is what I have investigated. And it's this one particular model is close to what's actually happening in the excised lung model that I have that they have. Anyway, that's, I don't know.
Yeah. Okay. Yeah. So how are you doing, Dick? What's going on there?
Oh, pretty good. We had a turkey dinner yesterday,
Okay.
so I'm sort of slow today.
Yeah. All right.
Yeah.
Yeah. So I guess we can talk about a few things today. I have an interesting paper on C. elegans cell fate, and then I have another thing on some things on the cell's operating system. It's kind of an interesting blog post, but we'll see.
Well, let's see if this is, if it'll turn on. Okay, slowly. Computer slow. I've got this explanation for how the stationary solver solved my equations. And it says compile equations stationary in study 1, solution 1. And then it gave the time it started, geometry, shapes, function, linear, Lagrange. And then it says running on Intel 64 family 6 model 158 stepping 10, genuine Intel. And then using one socket with 6 cores in total on desktop such and such. Anyway, what I wanted to know is what on earth do they mean by stepping 10?
Well, maybe it's like step size in the simulation.
Could be.
Uh,
What does that translate into seconds, for instance?
let's see. I'm not sure. I might look it up here. Usually the step size is like arbitrary units of time. There could be in the documentation like a mapping between seconds and units of time, but it's hard to say. Let's see. What is this? Are you using COMSOL?
Yes.
All right.
Yeah.
So it says controlling. Oh, okay, yeah. There's the okay. I found in the documentation controlling the time-dependent solver time steps. I don't know if that's what you're looking for, but basically they talk about like in the solver, it's it estimates an initial time step size it take based upon the total simulation time. Thus, if you've solved the same model twice for different total times, the initial time step can be different. This can lead to different results if there's something significant happening immediately after the start time. Then you can specify the initial time step size, go to the time step solver settings, time stepping section, enable the initial step option, and enter a fixed initial step size.
Okay, where did you look at that from? How did you?
Grab it. Yeah, I'll grab it here and put it in the chat.
Oh,
So
okay.
this is from the COMSOL documentation.
Oh, really? As
Yeah.
I looked for it.
Well, I just searched on I think Bing or something and came up with this. But I mean, this is like, yeah, it looks like there's a screenshot in there where they just talk about the time units. So like you can set the time units and the range, and then it has like it goes through from one point to another, but of course it takes a step size every time there's an iteration.
Okay, well, you came up with that faster than I certainly did, so
Well, I don't know
thank
if
you.
it's useful. Yeah, but I mean, it probably is. But like, yeah, step size is always kind of like arbitrary units, but you usually define it in the simulation, or usually you can define it in terms of a process. All it is just that iteration. Yeah, this is like the description you put in the chat. So like, let's see. So that yeah, it's just describing like what it's doing. Stepping 10. Why is it say genuine intel? I guess that's the thing that they yeah. That's what they accept.
Advertise.
Yeah. Okay. Yeah. So it's a linear Lagrange running an Intel 64 failure. Yeah. Okay. So I mean that's all like information you need because every computer is going to run it differently. We've been talking, of course. Morgan and I have been talking about a lot about virtual machines and running simulations of virtual machines. And the advantage there is that you can run them independent of the hardware. Like you can run them on different hardware and get the same result. But that's, you don't need to do that. It's just kind of one of these things we've been talking about.
Well, the stress strain curves of tissue are very dependent, and tensegrities are very dependent on the how the force is applied, whether it's fast step, slow step, continuous step, like a staircase step.
Yeah.
Like I don't know what it is. I don't know if it's a staircase or if it's a square wave. And what's the ramp on the leading edge of the square wave? I don't know. I can't tell from it. I don't know. So you ask, well, what's the rate here? Well, the computer says it's 10. Yeah, 10.
So yeah, I mean, I wonder if people have done like experiments in soft matter, like to look at some of these parameters. Like you can get, parameters from biological materials, but also from materials that have basically the same hardness and things like that. And you can just
Well,
see
there have been experiments. I've got examples of how the applied force rate changes the curve. I have that.
Okay. Yeah, I mean that's fine.
So I have a thesis with, I have no idea what the rate is of the applied force. It's a main feature of
Yeah.
oops. Anyway, so that's my complaint today, I guess, because it doesn't say.
Yeah,
Then it's 10.
yeah. Yeah, it's hard to get all these things sort of calibrated and figuring out what's like realistic and what's not. But
Yeah, and if you go into time stepping, like then they will give you ways of altering the leading edge of the curve for your experiments. But this is a stationary study, so no. There might be a way of altering stationary studies time step, but it's maybe I should ask COMSOL that.
yeah, you could do that.
But I just need to know what it is. At this point, it was done at this rate. I don't care.
Yeah.
I just need to know what it is. And 10 doesn't cut it in engineering.
Yeah. Well,
Literally.
I don't know what people are doing in other studies with COMSOL. If people are doing anything close to this, like I don't know what the
Well,
closest thing would be.
It's a building, and it doesn't matter. You hit it with a little hammer or a big hammer, it still sits there.
Yeah.
If you hit it with an earthquake, well, that's different.
Yeah.
Yeah.
Well, that would be a good paper. Earthquakes and embryos.
Yeah. Earthquakes and insegrities. They use
Yeah,
tensegrity modeling for buildings, for earthquakes,
right.
to make them earthquake proof.
Yeah.
They do.
You have to, yeah.
Anyway, yeah, so it's still, anyway, it doesn't solve the problem of what is 10.
Yeah.
So I have a 10 problem.
Yeah.
10 units. 10 units. What are the units?
Yeah.
Can I ask Susan a quick question?
Yeah.
Yeah, Susan, are you familiar with spherical tensegrity structures?
Sort of.
Sort of. Okay, there's the spherical cells. Okay, I have a question. If I designated N struts, okay, and put them on the surface of a sphere, and to calculate whether or not the structure is stable.
I could throw it in COMSOL. It would tell you immediately whether it was, how stable it was.
Oh, okay.
And there are some tensegrities that are not stable in COMSOL. There are quite a few of them. But if they are stable in COMSOL, they are definitely stable.
Oh,
But there's some structures that are not stable in COMSOL that in real life would stand up.
okay. Okay, good. For future reference.
Yeah.
Got a very strange project. I'm trying to determine whether or not tensegrity has any relationship to the structure of the atomic nucleus.
Oh yes, I was supposed to send you the Mathematica code, but it's 4 triangles.
Oh, okay. Well, the problem is in atomic nuclei, there seems to be what they call magic numbers, which make them particularly stable. And my question is whether or not there's something similar in tensegrity spheres. Okay, but it would be a big project because the number of struts would go up to I don't know, 100 or so.
Well, that's 100 is doable.
Oh yeah, 100 is doable, but 1 to 100,
One,
all of them.
okay, yeah. Like I said, it's not. You can't automate it. If you're using finite element analysis on a tensegrity, you probably can't automate it.
Oh,
You can't use their normal method of giving us a step, changing a step size in COMSOL because it wants to use the automated method to change the step size. And the whole thing falls apart.
oh, wonderful. So it becomes unstable even though it's stable.
Well, it doesn't give it enough time to settle. I think that's what it is. I think that's the problem.
Okay, gotcha. Okay, Bradley, carry on.
All right. Well, yeah, I guess that's about as far as I can go with helping you on that, I guess.
Okay, well, thanks.
Yeah.
If other, like I've asked this of a couple other people, and they have similar issues. So, okay, I'll give it to my professors this afternoon and tell them I've asked some people who are fairly knowledgeable about computer systems, and they don't know.
Yeah, well, I mean, yeah, it's probably a COMSOL specific thing, so I'm
Yeah.
not knowledgeable in COMSOL necessarily, but
Well, there it is. I mean, you found this section where they mention it, but anyway.
well, good luck with that. and hopefully things pan out.
Yeah, thank you.
All right. Sometimes they just tell you what they want to see. They don't care if it's like, you have to do a workaround and the committee has to decide what that is. So,
Yeah. Or I could send the whole quote from COMSOL to COMSOL and say, okay, so I have 10. What is that?
yeah.
I think I'll do that. Send them a help request.
Yeah.
They hate those, but that's okay.
Yeah. Okay, so the first thing I wanted to talk about was this article. It's a blog post that kind of goes, kind of rambles a little bit, but it's basically this uh, consideration of your operating system on a thumb drive. So they're talking here about uh, ribosomes and sort of the RNA associated with ribosomes and the genes associated with those ribosomes. So this is in bacterial. I think they're focusing on bacterial cells in microbiology, and then sort of thinking about that in terms of mammalian cells or your eukaryotic cells more generally. So this is by Christoph. This is, I think, the American Society for Microbiology blog. So there, it's a blog post. So starts off at the heart of every cell's operating system, you'll find the ribosomes. These ingenious nanomachines of about 20 nanometers in diameter that translate messenger RNA or mRNA into all kinds of proteins the cell needs to build and maintain itself. So if you know the biology, you have mRNA molecules that are basically transcripts from a gene. They get fed into this machinery. The mRNA are transcribed instead of translated at this stage to amino acids. Those amino acids build a chain which contribute to proteins. And so ribosomes are those sites where translation occurs. There's a very, highly sort of highly evolved mechanism of different types of tRNAs or translational RNAs, which can take those the codons from the mRNA and translate them into amino acids. And so, there are studies where they look at the different pools of tRNAs and how they, work to sort of transcribe some things over other things and that sort of thing. So there's not only translation going on at ribosomes, but selection of different codons and selection of different amino acids, and they contribute to proteins. And so the other thing to know about ribosomes is there are a lot of them in a cell. So it's not just one ribosome. There are a lot of ribosomes in different places in the cell, in the where mRNAs can be fed into this machine and turned into proteins. So this kind of goes on about this. See ribosomes busy at work here. This is another image where they show kind of I don't know if this is a good picture. This is just kind of some micrographs of E. coli chromosomes being transcribed and then simultaneously translated. And then this is a picture of the rRNA genes that are co-transcribed. So that's another blog post on that same blog. Ribosomes were first described by the electron microscopist George Palade in a paper from 1955, which he ponders on electron-dense particles lining the endoplasmic reticulum of rat pancreas cells. And then the quote is, the relationship between the membrane of the endoplasmic reticulum and the small granules deserves special consideration. Indeed, they deserved and got consideration. So we have at least 50,000 papers in PubMed dealing with ribosomes, and that's about the same number of ribosomes we have in E. coli cells. So these are bacterial cells, E. coli cells, and even just in bacterial cells we have 50,000. And in eukaryotic cells we have more than that. So this is a ubiquitous machine, I guess you could put it. With such numbers, it's no wonder cells spend roughly 2/3 of their energy budget on the synthesis of ribosomes and translation. So that's an interesting statistic that's really, impressive that the cell spending most of its, or maybe 2/3 of its energy on translation and synthesizing ribosomes. So, and of course it's important to make proteins, but it's a lot, so it's a really important process. And then he says, and yes, cells really tend to stick to their operating system. So I guess what he's referring to as the operating system are ribosomes and ribosomal RNA and tRNAs, translational RNAs, and the sort of the machinery that is involved in that. So organelles such as chloroplasts and mitochondria have ribosomes, and their genes encoding their components that resemble those of their bacterial ancestors. Well, your ribosome is basically information, and you can put it on a thumb drive, which is its whole other analogy. But anyways, continue on here, and then and arranged into subunits, and the subunits that are in there differ from prokaryotic cells and eukaryotic cells, with the sites of protein synthesis and mRNA binding at their interface. It was assumed for decades that the ribosomal RNA provides a scaffold on which the orderly bound ribosomal proteins perform their job of joining amino acids to a growing peptide chain. The first hint that this might be less than half the truth came from deciphering the genetic code, when it was shown that codon-anticodon interactions of ribosome-bound tRNAs, which are the translational RNAs, and mRNA, which is the messenger RNA, drive translation. So this is where we have these codon-anticodon interactions with proteins being casual bystanders. So this is where the model where you have the mRNA being read off is sort of these interactions, and then the proteins kind of being the result of that. But it still came to some surprise when the elusive ribosome peptidyltransferase, the enzymatic reaction that joins amino acids by a peptide bond, turned out more recently to be actually a ribosome type activity located in the rRNA moiety of the large ribosomal subunit. So we have a remnant of the RNA world as key component of the cell's operating system, maybe. So they're making this kind of statement about how the ribosome is doing maybe more than just kind of producing proteins, that the proteins are actually interacting with the ribosome itself. We have this sort of RNA world remnant where RNA was really the thing that was, involved in passing information on in terms of heredity and life instead of DNA. So intuitively, you would probably guess that in prokaryotes, the gene encoding such vital parts of the operating system are encoded on the chromosome or the hard drive. Again, another metaphor. They are in almost all known cases in archaea and bacteria alike. In bacteria, the three ribosomal RNAs, 16S, 23S, and 5S rRNAs, are encoded by rDNA genes that are organized as an operon and transcribed from a common promoter. So the operon is this sort of set of promoters and genes that are ubiquitous in the bacterial genome. And so they're talking about these rDNA genes. And you have an operon where you have multiple genes transcribed from a common promoter. And that's figure 3, which is down below. A long primary RNA transcript, which includes the two so-called internally transcribed spacers that in many cases encode tRNAs, is processed by specialized RNases, which are these enzymes, to yield the individual rRNA species that are then assembled with the ribosomal proteins into precursor ribosomal subunits. So this is figure 3 here. This is a schematic representation of sequence variation showing these different genes for these different subunits in an operon from 5' to 3' end. Okay. So kind of talks a little bit about how deviating from the rule that in compact bacterial genomes, most genes come in just one copy per genome, one finds between only one RNA operon. So in some species of bacteria, we have only one RNA operon, and this is for a genome size of 9.1 megabases. and a whopping 18 in Cyanobacterium, the species of Cyanobacterium, with a genome size of 10.6 megabases. So apparently the number of RNA genes is not related to a genome size, but may reflect different ecological strategies of the bacteria. Although mostly confined to a chromosomal localization, a few cases are known where additional copies of the RNA operon are located on large plasmids, which are present in all Vibrio species, in numerous alpha and beta proteobacteria. In many cases, provides another example for an operating system on a thumb drive. So this is, again, I guess where they're going with this is like there's this, can you describe the entire operating system of a bacterial cell? And then is it compact enough to fit on a thumb drive? And we're thinking in terms of what the genes that are encoding these. So there's an operon that encodes all of these RNA subunits. Then those subunits contribute to this mechanism. And that's the thing that, so we put all of that on a thumb drive. This is important for synthetic biology because we want to be able to build, maybe a minimal cell where we have a minimal set of components that can actually self-sustainingly transcribe genes, produce proteins, and do so maybe like some sort of container. And so that's kind of where they're going with this. So does this mean that also rRNA genes participate in horizontal gene transfer? There is some evidence that this is in fact the case. Therefore, rRNA-based phylogenetics, the marvelous idea that allowed Carl Woese to propose his 3 domains model, which of course was a famous study where Carl Woese laid out the framework for eukaryotes, prokaryotes, and archaebacteria, may turn out to be really messy in some niches at least. This would then be the normal state of affairs in most of biology. So it's an interesting post. Basically, the idea is you can take these rRNA genes, create ribosomes, and then those ribosomes, if you have some genome, you can transcribe that genome and produce proteins. And so how big does that need to be? And that's what they're kind of talking about. Okay. All right. So then the next thing I wanted to get to today was this paper. This is a bioRxiv preprint. This is a paper on early embryogenesis in C. elegans. It's by a Chinese group. And they're talking about quantitatively resolving cell fate in the early embryogenesis of C. elegans. So what they're going to do is they're going to try to figure out how cell fate is determined in early C. elegans. We know that, for example, it's a deterministic process, meaning that you start out with like a 2-cell embryo where you have basic polarity at an anterior and a posterior end. And those 2 cells produce 8 founder cells. And then those 8 founder cells go on to produce different types of tissue, different types of organs. And you can't really obliterate those early cells without basically creating a non-viable embryo. So you can't just have cells that kind of compensate for other cells not being there. C. elegans produces a certain number of cells for the adult, and if those cells are missing in the embryo, then they're missing from the adult, or at least they're sort of the descendants are missing from the adult. So what they're going to do is they're going to talk about cell fate and how we can resolve this quantitatively. So let me go into the abstract. So the abstract reads, the nematode C. elegans exhibits an invariant cell lineage during its development, where the gene molecular network that regulates the development is crucial for the biological process. Well, there are many molecular cell atlases describing the phenomena and key molecules involved in cell transformation. The underlying mechanisms from a systems biology perspective has received less attention. Based on an endogenous molecular cellular theory that relates the molecular mechanisms to biological phenotypes, we've constructed a model of core endogenous network to describe the early stages of embryonic development in the nematode. So what they're going to do is they're going to create a gene regulatory network model of these early stages, and they're going to pick candidate genes that are involved in this early process, and they're going to build a genetic regulatory network that shows how those things are sort of upregulated and downregulated in that network. So that's what they're doing here. Different cell types and intermediate cell states during development from zygotes to founder cells correspond to the steady states of the network as a nonlinear stochastic dynamical system. So the other thing they're doing is they're taking this and they're building a nonlinear stochastic dynamical system, and they're going to try to, that's how they're going to try to get some insight out of this. Connections between steady states from a topological landscape that encompasses known developmental lineage trajectories. So, we have these steady cell states and we have these transitory states or these transitions between states. And, sometimes you can treat that as sort of a Markov model where there's a certain probability of transition. But you can also treat it as this sort of nonlinear stochastic dynamical system that you can plot on this topological landscape, basically this differentiation landscape that allows you to describe known developmental lineage trajectories. By regulating the expression of agents in the network, we quantitatively simulated the effects of the Wnt signaling pathway on cell fate transitions and predicted the possible trajectories of transdifferentiation of the AB cell, which is one of these founder cells across the lineage. So they're going to look at the Wnt signaling pathways. They're going to look at that effect on cell fate transitions. The Wnt and Notch are sort of like at the top of the network, and then there are other genes that are below that are going to be affected by that activity. And then they're going to look at the different trajectories of transdifferentiation in the AB cell across the lineage. The success of the current study may help advance our understanding of the fundamental principles of developmental biology and cell fate determination. So they kind of talk about the importance of these early stages of development, and kind of how that is, C. elegans is an important model for that. They talk about the sources of single cell RNA sequencing data. So this is RNA-seq data that they're focused on, and how we can make really high definition maps of this process. So the single cell transcriptome of the L2 larval stage of C. elegans has been well characterized, along with the construction of cell maps at various stages of early embryonic development and the creation of spatiotemporal atlases. So this is all in different aspects of development. So of course we have the larval stage, L2 larval stage. It's been well characterized in terms of single cell transcriptomics. We also have some data from embryonic cells, but it's that's still kind of a work in progress. We have cell maps, so we have like the data. It's kind of in pieces, and so we can get access to some of that data to inform this process. So molecular structure generates vast amounts of information, but we have to sort of make sense of it. And so that's what they're going to do here. And then they talk about Waddington's landscape and how that might end up being this sort of way to kind of make sense of a lot of these data for different cells. So, during the developmental phase, cells reach their final differentiated types via distinct fate decisions. Exploring the changes in cell states in the process helps understand the relationship between different cell types. Waddington's epigenetic landscape conceptualizes developing cells as balls rolling down a hillside. And then they talk about like the minima and maxima of that landscape. The shape of these hillsides are determined by gene regulatory networks. And so you can work on gene regulatory networks for this process. And Murray et al., which is a data set that, actually the Murray 2012 data set is important for understanding what the key regulators of different lineage fates are. While Maduro et al. Established the gene regulatory network governing cell fate specification in the endomesoderm during the embryonic development. So this is where they kind of piece together a lot of the available data. Okay, so they talk about the founder cells, and then they kind of get into this sort of endogenous network modeling that they're doing. You have to pick a number of different genes for this. They actually want to uh, pick like 4 modules of functional genes. So they selected 4 key modules: cell polarity, cell cycle, signaling pathways, and lineage specification. So this is where you can do, go into the data, look at the genes, look at their function, or at least their annotated function, and you can construct networks based on this function. So, there are a number of genes that are involved in cell polarity. There are a number of genes involved in cell cycle, signaling pathways, and lineage specification. And those are all sort of the core parts of these modules. So we have 4 modules, and we presume they interact. There are genes within those modules, and then we want to look at how those modules interact. So when they talk about an agent here, they're talking about a node, which is basically just gene, and then its expression state is contingent on the expression state of other agents or nodes. And then, whether that relationship is supposed to be sort of suppressive or facilitative, whether it leads to upregulation or downregulation, then that's how the network will function. So an agent on the network in the current context can represent typically a cluster of relevant genes, molecules, and or a signaling pathway. The genes or proteins regulate each other directly or indirectly through transcription, translation, or signaling. Hence, we take a coarse-grained approach to abstract these regulations into activation or inhibition interactions. So it's like activation would be upregulation, inhibition would be downregulation. Okay, so then they talk about the polarity during asymmetric cell division being controlled by the par polarity proteins. And then you can map that to different genes that are involved in that protein. So we have PAR6, which is representing the anterior protein complex PAR6, PAR3, PKC3. Well, LGL1 represents the posterior complex LGL1, PAR1, and PAR2. So you have this set of proteins that represent the anterior and posterior ends. You have PAR6 and LGL1, and then those 2 genes represent this protein complex. Okay, and then PLK1 stands for the complex PLK1, MEX3, and MEX5, which links polarity protein to the asymmetric distribution of agents within the cytoplasm. So this is where we have the polarity genes, polarity proteins, and then the asymmetric distribution of different nodes or whatever within the cytoplasm. So the distribution within the cytoplasm and the basic polarity. Okay, so then the daughter of cells of AB divided more rapidly than those of P1. So basically, you can explain that polarity through these different activities in the transcriptome, leading to the production of proteins. So in this case, AB, which is the anterior end, is favored over the posterior end, P1, at least in terms of cell division, and how that asymmetry is being produced. CDC25 and CDK1 are chosen to represent the promoters of the cell cycle. So if we're talking about cell division, we're talking about cell cycle. Well, WEE 1.1 represents the inhibitors. So we want to have the promoters and inhibitors in this network. During the division of AB and EMS to generate daughter cells, so AB and EMS are 2 founder cells, 2 key intracellular signaling transducers are involved. GLP-1 receptor represents the Notch pathway, and TCF transcription factor POP1 represents the Wnt signaling pathway. So we have GLP-1, which is the Notch pathway, POP1, which is the Wnt signaling pathway. And so these are involved in growth and differentiation. This is, of course, across taxa. You see this in Drosophila, you see this in mammals. Okay. Key factors that determine cell differentiation include maternal transcription factors like SKN1 for mesoderm and endoderm specification, PI1 for germline cell fate, and PAL1 for the somatic descendants of the P2 blastomere. Additionally, lineage-specific factors include TBX35 and TBX37 in the mesoderm, PHA4 for the pharynx, HLH1 for muscle, N3 for intestinal endoderm, Lin26 for ectoderm, and so all those are sort of in this network as well. The above agents are listed in Table S1 in supplemental materials and in 1A. So this is actually our figure 1. This is the network. So you see that you have these different proteins, and they just described what the functions are. They're also color-coded here. So we have cell cycle in blue, signaling pathways in yellow. This is Wnt and Notch, respectively. We have cell polarity, par6, LGO1, PLK1, and then lineage specification, which are the things that specify these different lineages down here. And these connections, the red connections are inhibitory, the green ones are activation. So like, for example, if we take N3, if more N3 is produced and there's more N3 protein, it goes up to We 1.1 and it facilitates the production of We 1.1. But if We 1.1 is, if it's facilitated, then CDK1 is inhibited. If CDK1 is, if it's encouraged to proliferate, then WE 1.1 is inhibited. So you have these relationships in the network. And of course, when you have these kind of, like if we think about a neural circuit as well, it's the same thing. We can have these inhibitory relationships. We can have these reciprocal inhibitory relationships. We can have relationships where we have a positive feed-forward relationship and a sort of an inhibitory feedback relationship. So we can have a lot of interesting dynamics come out of these models. So that's figure 1A. Figure 1B just simply shows the Waddington landscape on the left. We see the sort of the phase diagram on the right. And we see that there's this potential. So potential is going upward, basically where we have cells that are in steady states, which are in blue, and some transitional state, which is in pink. And the idea is that on this energy landscape on the right, the steady states are in the wells, much like they're in the channels of the Waddington landscape. And then when they need to transition from one state to another, they need to climb this energy barrier to the top, reach this transitional state, and then drop back down to this new steady state, which is another energy well. And so, you need to have this sort of potential that rises to go from one state to the transitional state, and then a loss of potential to go back down to the new steady state. So there's this metaphor of hill climbing and descent, gradient descent, not the gradient descent we talk about in machine learning, but sort of related. It requires some sort of, maybe an energetic input, maybe a transcriptional input, something to drive it from one point to another. So that's your figure 1. It kind of describes what they're doing with this model. So those are the interactions of the genes and the gene regulatory network. And then they work on building this non-equilibrium stochastic dynamical model. So this is where we have these stochastic differential equations that they build. So the dynamics of n agents within the endogenous network, which are these nodes, with their level of expressions or activities denoted as a set of x, can be represented by a set of stochastic differential equations. So this is their stochastic differential equation, where you have this component that's force, which is this function here. The deterministic part is force, which is a generically nonlinear function, is the rate of change for the expression level of the xi of the ith agent. And you can also, and then the other part of that, it's additive. The other part of that is multiplicative Gaussian white noise. So that means that we have this multiplicative noise factor and this sort of force factor. The force factor being deterministic, the multiplicative Gaussian white noise being stochastic. And so you add those two together and you end up with your stochastic differential equation, where you're going through and it's generating sort of this force and the stochastic component for each time step. So there's some parameters in here where there's noise intensity, which stands for the average over the fluctuations. Dx denotes the diffusion matrix. And then finally, we have a term that's a Dirac delta function. And then this, such a general form of a stochastic differential equation be further decomposed in the following form. So you can also write it like this, which is a bit longer, where we have a friction matrix, which corresponds to dissipation. A transverse matrix corresponds to oscillations. There's a scalar function representing a generalized potential. And then there's normalized zero mean white noise. So you can, take this basic stochastic differential equation, which has a deterministic and a stochastic component, and you can break this down into specific types of things going on. So there might be oscillations, there might be dissipation, there might be this sort of potential that is, we don't describe it in terms of molecular biology, but it's in the equation, and then this normalized white noise, which is the stochastic component. So all of this is basically to say that there are a lot of ways we can model this gene expression, the outputs of this gene expression network. There are a lot of things that we can connect to the molecular function in the cell and describe with these stochastic differential equations. So, we're looking for steady state solutions. We're looking for these transitional states. And so a lot of this goes through the math. There's this fixed points of the ordinary differential equations here, nonlinear ordinary differential equations, where they kind of identify the fixed points of these. And then, of course, they're at the results where they have this endogenous network simulation. So this is where they build this core endogenous network with relevant modules and core factors involved and the interactions between them. It's a closed loop system where each agent is regulated by other agents, forming feedback loops with no pure upstream-downstream relationship. That's what we saw in figure 1A, which is this network with these kind of relationships that don't really form any sort of linear or sometimes even coherent set of relationships, but they produce this dynamical output. So then we can model the network dynamics as a set of nonlinear ODEs. We can also look at the Boolean dynamics, and then this is their sort of matrix of results. So this is the 10 steady states obtained by ordinary differential equations. So if I can zoom in on this, let's see if I can zoom in on this a little bit more. Okay, you can see that there is these steady states, these 10 steady states, S1 through S10 going right. And then these are I think the genes here in the network. So these are all the genes as the steady states. And then this is the relationship. I guess, I don't know if it's correlational or what. Let's see. I think they're just expression levels. But the point is they go from 0 to 1. So you can see that there's, we have this heat map here from 0 to 1. 0 is dark blue, 1 is dark red. So you see that there's, CDK1, for example, has a lot of 1 values or 0.99 values for the different steady states. Then you have some, steady states that have a lot of inactivity of genes. Some steady states that have a lot of high activity of genes. So it's interesting, some of these steady states, what they're kind of coming out with. And then we have this other one where we have these Boolean solutions with the same thing. So you have par6, you have these same genes here, and they have 0 to 1, and then some of them aren't color-coded for some reason. Well, I think they're at 0.5, so they're white. That's how this works. And so you see a lot of sort of these Boolean states where you have sort of a similar distribution of things going on. Then you have these, let's see, the T is, zoom back out. So A is the 10 steady states obtained by ordinary differential equations. B are the 33 transition states identified by ODE. So the Boolean states are the transition states. Actually, no, the bottom one is B. So that is the one where we have the 33 transitional states. We have T1 on the left, T33 to the right. And so we have those solutions for this. And then this top one are the steady, just another way to obtain the steady states. And then C, okay, now C is the top here, never mind. Those are the 10-point attractors and 8 linear attractors based on Boolean dynamics. So you have 10-point attractors and 8 linear attractors. They're just different ways in which they are stable. Red colors in the grids mark high expression and blue for low expression. Okay, so then they did that. Then they had the phenotypes identified with experimental data. This is where they went and they looked at these steady states and they tried to find these in experimental data. So they were able to identify, let's see, so they had both S1 and S4 steady states exhibiting high expression of the posterior factor LGL1 and the germline factor Pi1, indicating their correspondence to the P lineage cells. And so in S1, we also see cell cycle promoting factors that are highly expressed, while the levels of inhibitory risk factors remain low. So we can actually identify this in P1 to P4 cells, S1, and then we know that that's true because they continuously divide with stem cell characteristics during early development. While the S4, which is the S4 steady state, corresponds to Z2, Z3 cells, which are a different part of the germline that enter a quiescent state during late embryonic development. In S3, S5, and S8, which are all steady states 3, 5, and 8, the endomesoderm activator SKN1 is highly expressed. Additionally, the mesodermal factor TBX35 is highly expressed in steady state 3. Therefore, it can be determined that steady state 3 corresponds to the MS cell, the founder cell, one of the founder cells, that mainly divides to generate mesoderm. The endodermal factor N3 is highly expressed in both steady state 3 and steady state 8. Considering the transition pathways between steady states, only steady state 5 can be directly converted from S1, representing the P lineage, through a single transition state. Thus, S5 is identified as the EMS cell, and S8 is identified as the E cell, which develops into the intestine. So what they're doing is they're looking at these founder cells, they're looking at these trends in expression, they're looking at the steady states that they found, and they're mapping the steady states to the founder cells. That's basically what they're doing. So they have 8 steady states that they're looking at, they map them to each one to a founder cell, and then they say, okay, these founder cells are producing something downstream. So some in MS, for example, that founder cell, which is in the posterior end of the worm, so that starts with this posterior asymmetry, that's going to mainly generate to divide to generate mesoderm. So we can actually identify from these steady states the appropriate founder cell, and then from that we can say something about the downstream effects of that. That's basically the logic here. So a lot of simulation, a lot of like getting these steady states, mapping them to these founder cells, and then validating them with high throughput data. So they use some data sets from GenBank GSE 77944 and GSA 83523. So these are transcriptomic data sets where they look at single cell transcriptome expression developing from the 1-cell stage to the 16 and 102 cell stages, respectively. So these are just kind of like going through the data sets and mapping this. So this is actually a very similar exercise to what we did in the differentiation waves paper, where we looked at different candidate cells or candidate genes. We didn't look at single cell expression too much, but we looked at this across the embryo and then mapping this to proteins and then saying something about, you know, in this case it was like the prevalence of certain, you know, genes in the developmental stage and then mapping that to a larger set of statements. Here they're looking more internally at C. elegans, and they're trying to really get a handle on some of these changes in the lineage tree and how, you know, how they can interpret that. So that's good. That's a lot of the work that they did there. They also have this low throughput data here. It kind of gives, you know, similar results to what they did in the first figure. And then they talk about how that maps to an epigenetic landscape. And so this is quantitative realization of Waddington's epigenetic landscape, kind of like going through 10 different cell types. So it's the 8 founder cells and then 2 additional cells where you have A, B, and Z, I think, are the 2 additional cells, in addition to the 8 founder cells. And looking at the possible switching pathways between cell types, which allow these to be connected by these transition states. So all these 8 founder cells are connected through the lineage tree, and we want to find these ways in which they are transitioning between one another. So they also kind of start with P0, where the egg is fertilized, and then there's this 2-cell stage where you get these initial asymmetries, and then you go to the 8-cell stage where you get these founder cells, and they all kind of have to switch their program from the 1 cell down to the 8 cell. And so the question is, what are these switching pathways? What are these transitional states? Okay, so then they find that there are 24 developmental pathways between early embryonic development, 9 of which align with experimentally observed phenomena. So that's an interesting paper. I think that's focusing on the lineage tree and the early lineage tree. So I think that's well characterized. Getting down past that might be a bit of a challenge. I think their work is interesting, especially the dynamical modeling that they do. Dick, were you going to say something?
All right. Yeah, could you put the reference in the chat, please?
Yeah, let me go back. Get the DOI here for this. I'll just pull up the link here. And this is Quantitative Resolving Cell Fate, the Early Embryogenesis of C. elegans. There it is. Okay, yeah, so that's that paper.
I hope I have better news next week.
Yeah.
So, I've handed the whole thing in.
Oh,
This
good.
is the last little bit, like the validation of my model and can it be used for other situations.
Yeah, yeah. Yeah, that's good. I think that'll be interesting to see what your committee says. And, I know you're kind of working like in a new area, like you're working on tensegrities, and this is sort of biotensegrities, and of course, there are things that you can't easily translate that don't
Yeah,
really translate. So
you're running into roadblocks every time you turn around. It seems
yeah,
like this morning's. Oh, what is 10?
yeah,
Sure. The time is 10. Yeah.
yeah. All right. Well, thanks for meeting. See you around next week.
Right.
All right. Thanks. Take care. Bye.
All right. Bye.